Hello, everybody. Notice we are really running slow with the photosynthesis, and we still have to move on to respiration. So I decided to make this complete video for photosynthesis so that we can move on to the next lesson in the class. So we talked about photosystem 2 in the class, and I mentioned to you it consists of pigment complex and an electron acceptors. So it receives electron when the water is being splitted, and also oxygen is being released as a gas. So now the electron transport chain is being produced, whereby all the electrons being collected is being gathered. So, and this consists of cytochrome complexes and plastoquinone. These are enzymes which are very important in the process of photosystem 2. Cytochrome complex is an enzyme found in the thylakoid membrane of photosynthetic organisms. They help to catalyze the transfer of electron from plastoquinone to plastocyanine. So the prostacyanine cyanine is where the electrons are being gathered into. So mind you, there is carries electron from photosystem 2 to 1. You know, I mentioned to you before, the photosystem 2 produces the electrons, so which are sent to photosystem 1. So, and also it pumps hydrogen ion, which is produced when the water was being splitted. It pumps that into the stroma. It pumps that from the stroma into the thylakoid space. So, you know, I mentioned to you before, the photosystem 2 starts in the thylakoid. So the completion is in the stroma. So it's got a pigment complex and electron acceptors. So adjacent to enzymes that reduce NAP, NADPH, NADP plus to NADPH. So I mentioned to you the process in which this has been reduced. So when you have two molecules of two electrons and one molecule of hydrogen ion, when they combine together with the NADP ion, they form NADPH, which is a source of energy. So mind you, I actually want to show you how we can make up the reaction. So it's not difficult. You have to have NADP plus. So you have NADP plus. So you have NADP plus and plus 2 electrons and plus hydrogen ion. So that gives you that gives you NADPH. So this is what it gives here. So this is how we do it. So, and the next thing is a ATP synthase complex. So what is the function of ATP synthase complex? This is what generates ATP. So it has a channel for the hydrogen ion flow. So this synthase complex has a channel for the hydrogen ion flow. So the hydrogen ion flows through the channel and drives the ATP synthase to join the ADP. So when the ADP synthase joins the ADP and P, it forms a what? It forms ATP. So this is exactly what causes the production of ATP molecules. Now this is an example of it's an example of how it happens. So we've got a cyclic pathways for photosynthesis. So energy level, when it increases, so what happens is electrons, the photosystem 1, this is the pigment complex. The meaning is when we have a collaboration or collection of pigments. So then this is a reaction center when the sun strikes it. So electrons have been released and they've been sent to the electron acceptor. So once the electron acceptor is gathered, a lot of these electrons, it sends them down back to the world through electron transport chain back into the system. So this is why it's being said to be cyclic. It returns back all the electrons and to the photosystem one. So mind you, ATP has been produced and it's been used in the Calvin cycle for the reaction and other enzymatic reactions that happen in the process of production of glucose, which are carbohydrates. So now we want to talk about the organization of the thylakoid. So among the things I've mentioned to you before in the class is that what happens is once the solar energy strikes the thylakoids, then hydrogen water is being passed through it. So what happens is the thylakoid membrane inside the reaction is called the light reaction, whereby it splits the water molecule and O2 is being released as a product. And also CO2 is being also used in the reaction and it produces carbohydrate, which is a reduction. So here, water is oxidized to produce O2 and carbon dioxide is being reduced to produce carbohydrate. So that is happening in the thylakoid membrane. So what happens is the thylakoid is being divided into two. We have a space and we have a membrane, which is the cover of the thylakoid. So now what happens when the water is being broken down, so that sends down into the thylakoid area, the space in the thylakoid, it sends a lot of hydrogen ion. It continues to gather up a lot of it. So during the process of ATP synthase, the complex leads to the release of hydrogen ion. They've been released to be part of the materials which is being used to produce ATP. So here we've got phosphate molecule with ADP and being energized by these to call this chemiosmosis. So using the word is a kind of osmosis reaction where chemicals are being involved in it. And now this is the way in which an electron goes down the transport chain until they are being used to produce the NADPH from NADP plus. So this is called NADP reductase. It's actually the one that reduces the NADP plus to produce NADPH. Now let's move on to the next slide. Now ATP production. So the thylakoid space acts as a reservoir for hydrogen ion. So the thylakoid space, which we saw, helps to reserve a lot of these hydrogen ions produced when the water is being splitted in the light reaction. So each time the water is being oxidized, 2 hydrogen ion will remain in the thylakoid space. So transfer of electron in the electron transport chain yields energy. We saw that. So this energy is used to pump hydrogen ion across the thylakoid membrane. So what happens is the protons would move from the stroma into the thylakoid space. So the flow of the hydrogen ion back across the thylakoid membrane would help to energize ATP synthase. So ATP synthase would enzymatically produce ATP from ADP plus phosphate. So the method of producing, this is a definition you need to know. This method of producing ATP is called chemiosmosis. Because why? ATP production is tied to the establishment of hydrogen ion gradient. So because we want to produce ATP, it has to be that until the hydrogen ion gradient increases, that will lead to the production of ATP. All right, so now let's move on to plants. How do they actually fix carbon? There's a cyclical series of reaction that takes place in this reaction. And these things that usually take place, they utilize atmospheric carbon dioxide to produce carbohydrate. So what we know is known as C3 photosynthesis. I'll talk about this when we have a slide. So what happens is there is a way in which carbon dioxide is being used to produce carbohydrate. But we know that C3 is actually that the six carbon atoms is being splitted into two. So when that happens, it involves three stages in this reaction. So the first one is called the carbon dioxide fixation. So carbon dioxide is being fixed into the reaction. So after fixing it, then the carbon dioxide is being reduced. So that is a reduction stage. So after the reduction stage, the carbon dioxide is not being used to generate RUBP. So RUBP is called ribulose. RUBP is called ribulose diphosphate. So let me write it down for you. It's called ribulose 1,2 phosphates. So let me call them pen. So we need to know, we can write this down somewhere. So it's called ribulose 1,5 biphosphate. So it's called ribulose or ribulose. So ribulose 1,5 biphosphate. It's a bisphosphate. So let me explain this to you a little bit. This RU stands for ribulose or ribulose. So the phosphate, the BP is bi, and phosphate, the P. So the 1 and the 5 shows you the where on the carbon atom where they attach to. So that's what gives you the 1,5. So hopefully I will find the chemical bond thing and I will show it to you. So now let's talk about the fixation of carbon dioxide. So the CO2 is attached to 5 carbon. So I want you to understand this concept very well. We have a 5 carbon of this one, to the 1,5 bis phosphate. So the 5 carbon, which is RUBP, which is a ribulose 1,5 bis phosphate. So the CO2 attached to it, and to produce what? To produce 6 carbon molecules. So it's been facilitated by this enzyme called RUBP carboxylase. So what happens is it produces 6 carbon atoms or carbon molecules. So this is now being split into 2 3 carbon molecules, which is called 3PG. 3PG is called 3-phosphoglycerate. It's called what? It's called 3PG. It's called 3-phosphoglycerate. Sometimes it's been called 3-phosphoglyceric acid. Either of the ways you pronounce it, the one which is either, no one's going to ask you to write this in the exam. Just write 3PG. It's okay. So it's being called 3-phosphoglycerate, or you call it 3-phosphoglyceric acid. So then what happens afterwards? The reaction has been accelerated by RUBP carboxylase, which is called rubisco. So this rubisco is an enzyme which helps to what? Which helps to facilitate or accelerate the reaction. So what happens afterward? The CO2 is now being fixed. So because it is part of the carbohydrate. So you fix this. So that was what we meant by fixation of carbon dioxide. Because you cannot work with the 5 carbon, you need to have one more to make it 6. So this is what fixation is. Now let's go to the Calvin cycle. Now, in Calvin cycle reaction, what usually happens is after all this process, which I already explained to you before. So what happened is the 3 CO2 molecules is being added to the reaction, is being fixed, which is called carbon fixation. So it's been combined with an intermediate, which is 3C5. So the intermediate is just part of the reaction that produces what you want to produce. So after that process, so what happens is after the process, of dividing the carbon into two 3-PGs, which is 3-phosphoglycerate. So what would happen is it's going to produce 6 ATPs, which will be combined to 6 ADP to produce ATP. So, and after that, so now you're going to produce what? You're going to produce 6 BPG. So the BPG is the what is 1,3 bisphosphoglycerate. It's 1,3 bisphosphoglycerate. So that will be produced afterwards. Then notice the ATP and the NADPH molecules were produced by the light reaction. I think you still remember that. So this is why it's telling you here, 6 NADPH has been what has been utilized and has been oxidized to produce this. ATP has been also used to produce this. So after that process, what happens? Then it produces glucose. So 6G3P, which is glycerate 3-phosphate, is going to produce for you a glucose molecule. So then other organic molecules are being produced as well. So then after that process, then it's going to return back to the 5G3P, which is in the beginning of the reaction. So then in this case, the 3-ATP is actually being, so these ATP molecules which we're producing in the light reaction are also being used in this process to produce the what? To produce 3-ADP plus 3-phosphate. So the meaning is in the carbon cycle, what we actually, the processes involves you having to fix the CO2. So because you need to have one more carbon molecules to have 6 carbon molecules. After you fix it, then it's been splitted into what? Into 3PG. So after the splitting, so the splitting will not produce what? 6 BPG, which is 1,3-bisphosphoglycerate. So after that, then it produces what? It produces the glucose, which you want to have. Mind you, it's going to multiply this by 2. It's going to produce double of it. So then after that, we have the 5GP3 being produced from it. Then after the process, it goes back to the reactor you started with. So this is why it's being called a cyclic reaction. If you want to have more information about it, you can decide to click on the alternate information on the slide, and you will see more information about it. So the second stage of the reaction is the 3PG is being reduced to BPG. So the 3PG is being reduced to BPG, which is 1,3-bisphosphate glycerate. So BPG is then being reduced to G3P, which is glyceraldehyde 3-phosphate. So electrons and energy are being required for this stage to happen. So this stage would now utilize the NADPH, which you produce, and ATP, which is being produced in the light reaction to produce more glucose. So G3P is reduced and chemically able to store more energy and form of larger organic molecules such as glucose. So this is a reduction of carbon dioxide. This is how it happens. So you would see the 3PG, which is 3-phosphoglycerate, uses ATP, which has been produced to produce BPG, which is 1,3-bisphosphoglycerate. So then after that, it uses the NADPH to produce glyceraldehyde 3-phosphate. So someone could ask you, which of this reaction actually uses ATP? It is the conversion of 3PG to BPG that uses ATP, and the conversion of BPG to G3P is the one that actually uses the NADPH, and it produces NAD+. So the third stage is a generation of RUBP. Okay, so as I was saying, I was explaining what actually happened here. So in this case, where we have the carbon atom, which is the carbon dioxide molecules enters the carbon cycle from here. This is how it enters. So it enters the cycle, which is 3 it becomes three glucose molecules. So this is a section where they fix the carbon. So what happens is being transformed to six molecules of three phosphoglyceric acid. So three, or you can call it three molecules of three phosphoglycerate. So with the what would they say? So sub three. So this is where ATP, six ATPs is been converted to six ADP. The meaning is it's been used. So when you say been converted to ADP means the energy has been utilized in it. So mind you, this ATP and the NADPH molecules were all produced in the light phase of the reaction. So after that, then we've got six molecules of three phosphoglyceric acid, or you call it 1,3-bis phosphoglycerate. So it's got three glucose molecule, we call it triose. It's been converted into six molecules of 2,3-base phosphoglyceric acid. So it becomes G3P. G3P is glyceraldehyde 3-phosphate. So at this point, it's got three glucose molecules, as we already mentioned here before. So what happens, because here we had six when this was fixed, but this case it got reduced and been splitted into three. The six carbon molecules have been splitted into three. So what happened is after that, so it leads to having a six. It leads to having a six molecules of glyceraldehyde here. How did it happen? Because when you produce one glycerol, we will produce glyceraldehyde 3-phosphate 2 times, so it's going to produce six molecules of it. So six molecules of glyceraldehyde 3-phosphate is being produced. So with three ions glucose, which is leading to five molecules of glyceraldehyde 3-phosphate here. So with thiose glucose, so mind you, the ATP is being converted here also to three ADP plus phosphate molecules, meaning that the ATP is being used as well. So mind you, all of this ATP we talked about here and NADPH were produced in the light phase of the reaction. So finally, what happens is three molecules of ribulose 1,5 bisphosphate is being produced, which with a pentose glucose, pentose sugar. Pentose sugar means a glucose with three molecules, three carbon molecules. So this is why you have C5. So this is a very it's a very complex system actually, but we just have to know that in the reduction of carbon dioxide, the 3PG is being reduced to BPG. So the 3PG is being reduced to 3PG. So, and we need to understand as well that the BPG is then reduced also to G3P. So when these processes are happening, what happens is electrons and energy are required for this stage. So this stage will utilize the NADPH and some ATP is produced in the light phase reaction. So this is G3P is reduced and chemically able to store more energy and form a larger organic molecule such as glucose. So now let's to this point. So we should understand, I was talking about this before, during the 3PG conversion to BPG, this is when ATP is being utilized, and during the stage of BPG, it been changed to GP3. This is when NADPH has been used and becomes NADP plus. So now in this case here, so what happens is the ribulose bisphosphate, 1,5 bisphosphate, has been used in CO2 fixation. It has to be replaced back. So because you use it, and that's why we call it cyclic. So every 3 turns of carbon cycle will produce 5G3P. Every 3 turns of carbon cycle produces a 3 carbon molecule. Also, uses the 3 carbon molecules to remake the 3 ribulose bisphosphate. So what do I mean? That is to say, every time, if you have a carbon cycle one time, 2 times, 3 times. So 3 times produces what? Produces 5 G3P. Uses it to make ribulose. So this is exactly what it says here. 5 times 3 equals 3 times 5. Because mind you, you need to understand this concept here. So when you have carbon cycle 3 times, it uses what? It uses 5 days of this. So then it uses that to remake this. So it uses that to remake 3 ribulose bisphosphate, which is a 5 carbon molecule, which you saw as C5. So this is exactly what we are trying to say there. So this is 5G3P. So it produces, it produces 3 RUBP. So 3 ATP is being used. So as 5 molecules of G3P becomes 3 molecules of ribulose bisphosphate. So 3 molecules of ATP becomes 3 molecules of this. So the same thing is happening. So in the process of changing these 5 molecules of this to 3 molecules of this, 3 molecules of this becomes 3 molecules of this. So this is called regeneration of ribulose bisphosphate, 1,5-bisphosphate. So now the next one is the importance of carbon cycle. So carbon cycle is very important in many things. As we see, if carbon cycle is not there, there will be no, then the main production of carbohydrate, which you were aiming at, will not be possible. So this is why it says G3P, which is glycerol 3 phosphate, can be converted to many other molecules as well. So the hydrogen and the hydrocarbon, which is, the hydrocarbon skeleton of the G3P can form a fatty acid and glycerol to make plant oil. It can also form glucose phosphate, which is a simple sugar. It can also form a fructose, which is a glucose, which is got combined, which is a glucose from fruits. So which is with glucose equals sucrose. So starch also and also it can form a cellulose. It can also form amino acid. So we're gonna see from this picture how all of these things are possible. So this is called the fate of G3P. So G3P can be used to form glucose phosphate. It can produce fatty acid synthesis. It can be used to produce fatty acid. It can be also used to produce amino acid. So when you combine that with fructose phosphate, it can form a sucrose. So it can form sucrose. This is why you see some fruits are very delicious, or some leaves. So it can also form a starch in root and seed. It can also form a cellulose, which is found in the trunk of the trees. That makes it very hard when you say it. So, and as you all know, the glucose phosphate will lead to starch in the root in the seed, and it is the cellulose that will form the trunk. If you've ever cut the branch, the bark of a tree before, it contains cellulose, and this is the reason why omnivores, we don't have enough enough enzymes to break them down, but the animals have it, most especially the herbivores. So now all the types of photosynthesis which can happen is the majority of plants, the majority of plants usually carry out C3 photosynthesis. So many plants usually carry out C3 photosynthesis. So this uses the ribulose bisphosphate, 1,5-bisphosphate, ribulose 1,5-bisphosphate carbonate, which is an enzyme, to fix CO2 to ribulose bisphosphate in the mesophyll cells. Mind you, we talked about mesophyll. It is a place between the leaf where all of the processes of photosynthesis usually take place. Now, let's say, for example, we have a hot and dry climate. What usually happens in those cases, we know normally from adaptation, the stomata must close to avoid wilting, to avoid wilting, which means loss of water. CO2 would decrease and more O2 would increase. The meaning is the CO2 would decrease. Why? Because it does not want to waste the resources to produce more of glucose, but it's going to be continuing with respiration. So this is why O2 would increase. So now O2 will combine with a ribulose with the ribulose bisphosphate leading to the production of CO2. So this is called photorespiration. So this is called photorespiration. So the meaning is respiration also. So what about C4 plants? The C4 plants solve the problem of photorespiration by fixing CO2 to PEP. So they fix CO2 to PEP. What is PEP? Is a C3 molecule, a carbon 3 molecule. So it results to what we call oxaloacetate. Oxaloacetate is a C4 molecule. So mind you, CO2, the carbon there is what is needed. The O2 is going to escape, but this carbon is what combines with the C3 to form a C4. So in hot and dry climates, the C4 plants would avoid photorespiration. So they would avoid it because they want to keep, they want to reserve or manage the resources. So the net productivity is about 2 to 3 times greater than C3 plants. So, and in cool, moist environment, C4 plants cannot compete with the C3 plants. So in a cool and moist environment, the C4 plants would not be able to compete with the C3 plants. So this is just a summary on this page. So let's talk about the leaf structure of C3 and C4. Let's see the reason why they have those qualities. C3 plants, we call them actually non-uniform plants. They have a non-uniform pattern of the mesophyll. So you will see the mesophyll is not in a uniform pattern. Look at the mesophyll being arranged not in orderly way for the C4. So it's visible, and as you can see, with circular bundles of sheet cells, black circular veins and stroma opening between the epidermis. So all of these things are qualities which you see with the C3. It's got bundle sheet here, it's got the veins, it's got stroma. So all of these are mesophyll cells. But in the C4 plants, it's got a uniform, it looks like a flower, pattern of the mesophyll is been with the circular bundles sheet cell. So it's got black circular veins within it and the stroma opening between the epidermis cells. So this is what makes it different from each other. So let's now take a look at how they use that, what they have to fix the life or make them to be able to cope. So the ribulose bisphosphate, so the CO2 enters into the carbon cycle system, and we know what it does. It helps to fix the oxygen, fix the carbon, so to produce 6 carbon atoms, which is later splitted in the 3PG section, which later produces GP3 and returns back the ribulose to the system. So, and also in the C4 plant, so for example, we've got mesophyll cells, the bundle sheet cells. So the carbon cycle produces a G3P, So now let's talk about the CAM photosynthesis. Crassulaceans acid metabolism. CAM plants partitioning carbon fixation by time. So CAM plant, they divide the carbon fixation by time. So what happens on the night, the CAM plant fix CO2 and they form C4 molecule, which is stored in large vacuums. During the daylight, NADPH and ATP are available. So what happened, the stomata are closed for water conservation. So the C4 molecule would release CO2 to carbon cycle. Now I want you to look at the difference now. In the CAM plant partitioning, they fix carbon in different ways. During the night, the CAM plant will fix CO2. So it would fixing CO2 means adding CO2 to the system. So it forms a C4 molecule because it's got C3 before. It adds one C to it to become C4, which is stored in the large vacuums. But during the day, it's got energy sources. So what happens is the stomata will close. It's not going to take any CO2 from anywhere, wherever. So no CO2 comes in. So the C4 molecule release CO2 to carbon cycle. So the C4 molecules, which is already produced, will release CO2 to the carbon cycle. Now let's see an example of how CO2 fixation is happening in CAM plants. Example of CAM plant is pineapple. So what happens is in the nighttime, the CO2 is being sent into the system, which has got CO3 to produce CO4. So the CO4 produces what CO2 and it sends that into the carbon cycle to produce G3P, which is going to produce carbohydrate. So, but in the C4 is not the same. So now let's talk about photosynthesis and adaptation in the environment. The different methods of photosynthesis each have advantages and disadvantages, as you know. So it depends on the climate. C4 plants most adapted to high light intensities. They are adapted to high light intensities because they're known to grow in dry places. They have they adapted to high temperatures. They have limited rainfall. While the C3 plants are better adapted to cold environment, high moisture. So the CAM plants are better adapted to extreme aridity. So they adapted to extreme aridity. CAM occurs in 23 families of flowering plants. They also found among the not flowering plants. So you can read more about the rest from your textbook. So this will be the end of the photosynthesis, and we'll move on to respiration, hopefully by tomorrow. Thank you very much for listening.